Cell life cycle, mitosis, meiosis is easier too. Cell life cycle, we have interphase, and this is when all the normal activity of the cell occurs. All right? There is no, I learned this in high school. My teacher told me that interphase is the resting stage of the cell. I don't even know when a cell is resting. A cell doesn't rest. It's always making proteins. It's always doing something. I've never seen a cell in a vegetative state. So interphase is not a resting state. Interphase is doing all its normal activities, except not dividing. Okay? So it's going to be all the different things going on. I'll show you that. Mitosis is the time where we're going to make one cell into two cells. All right? Cytokinesis is the last part of mitosis, and mitosis is going to make one cell into two cells, but it's going to separate the nucleus and its chromosomes to do that. But when we have the division of the cytoplasm, we call that cytokinesis. It's a part of mitosis. It's the last phase of mitosis, but they have a special name for it. It's when the cytoplasm gets split. So this is interphase. We break that up into three things. If this is the life cycle of a particular cell, and every cell has different life cycles, timing. One can last a few hours, one can last 70 years. All right, so it depends on what we're talking about. But let's look at a run-of-the-mill kind of thing. Most time is this green area, and that's interphase. Most of the time it's going to be an interphase. A very small time, this is let's say 1 hour. We're looking at maybe 10 minutes of the hour is going to go through mitosis. But 50 minutes is going to spend in interphase. In interphase, you have that broken down to three things. G1 is growth. That's going to be where the organelles are going to duplicate. S is where DNA is going to replicate. This is in preparation of doing mitosis. G2 is when it's doing all its normal activities making proteins. Mitosis is the cell division, and we have four phases that go on here. Prophase, metaphase, anaphase, telophase. Okay, and we'll talk about those. So this is just a close-up of the cell cycle. Okay? Now, mitosis. When it's time for us to have a cell to divide, the DNA needs to replicate, and that happens during interphase. It needs to make a duplication of its cells. You can't just take the cells and just split it, because then you're only going to have half the cells in the remaining, in the fate of the two cells that we're going to make. So you got to duplicate it, because this cell, it has to look exactly like this cell and exactly like this cell. So you got the duplication going on, and that's when you're going to be looking at chromosomes the way we see chromosomes, right? Then it's going to go through mitosis. It's going to split these things, and these two cells are identical to each other, and each of these cells is identical to this cell. We call this the parent cell. We call these the daughter cells. This mitosis, or if it goes to mitosis, it can happen, like I said, in a few minutes, or it can happen in 70 years. The neurons, the nerve cells, take 70 years to multiply. In most of the cases, you don't even multiply because you'll be dead by the time that happens. Okay? So, let me show you what's going on with mitosis. Now, I don't know if this is a good thing for you guys to draw what I'm doing, because the only way for me to explain this is I'm going to draw it, but I'm going to erase things as I go along. It's my animation. So I start doing this, and I erase things, and I usually have a lot of profanity coming out, like, he's telling me to erase this stuff. So just giving you a heads up, all right? So this is what happens. Okay? The cell and the nucleus. And you're going to have centriole over here. Remember, they're going to be close to the nuclear membrane. And right now, it's all chromatin in here. So you can't see anything. So this is interphase. Now it's going to go through mitosis. And the first phase of mitosis is that this chromatin is going to condense on itself. From all the sheets of pages that are on the floor here, we're now going to bound them back up into its books that go back on the shelf. And what you're going to have here and there will be 46 pairs there. I'm not going to draw 46 pairs, but you get the idea. Okay? So they're going to condense into chromosomes. So the chromatin now becomes chromosomes. They condense. Meanwhile, the centrioles are going to move to either side of the nucleus. So now they're going to go here and here. And the nuclear membrane starts to disappear. This is all during prophase. And what happens here is that the centrioles over here are going to start developing what we call spindles on either side here, attaching to the chromosomes. Okay? Okay? Questions so far? Alright, so what's happening here, just to show you on the picture here, here's the nucleus. There's chromatin in here. You can barely see the chromosomes because they're not chromosomes yet. You got the pairs of centrioles over here. Okay? What's going to happen is the nuclear membrane is starting to break up. The centrioles are going to move from one side, and one of them are going to go on either side here. And when it does, the nuclear membrane's gone. You've got the chromosomes all there. The spindle fibers are going on there. This is more or less like a marionette puppet. You know, like Pinocchio, they have the handle up here and the strings come down to the puppet. I got no strings to hold me, right? That you know what I'm talking about. So you got the handle here and you got these strings. The handles are centrioles. The spindle fibers are the asters. They're also called asters. They're the strings, and the chromosomes is the actual puppet. And you have control about it. But what's going to happen here is that they're going to eventually be pulling this. So now these chromosomes are going to start being pulled to either side, but it's like a tug of war. And what happens here is that they kind of line up all in the middle because that's where they're all going to be like in a tug of war. They can't go anywhere. Like think of it on a imaginary disc. They all line up there. Does that make sense? Okay? They're all in the middle. This phase, this is called metaphase. When they're all lined up in the middle there. Okay? Then what happens is anaphase. Now these are pairs of chromosomes. So now the pairs, the single pairs, are now going to be pulled to either side. They're going to break off from each other. And they kind of go this way, this way, this way, this way, this way, and the other ones go this way. They're being pulled to either side. It's kind of like, I know, it'll look like this. I'll do this for you guys. It's like if there was a string or a rope that was tied to me over here to a boat, and the boat pulls me, and my arms are like the arms of the chromosomes, I kind of go like this. Right? If it pulls over here, my arms go like this. So it makes these V's going out that way. Does that make sense? It's being pulled that way. Then you've got telophase. What's going to happen here is a few different things. The whole cell is going to start pinching off at this point. And there's also something called a furrow over here, which is not quite separated, but it's going to be very soon. We call that a furrow. What will happen here is that this is going to be pulled over to this side here, and on this side over here, and the nucleus is going to start forming on either side here, and the chromosomes are going to turn back into chromatin, and the centrioles are going to be back on one side over here. And now we're going to also have the division of cytoplasm on either cell, called cytokinesis. So here's where they're all lined up in metaphase. This is where they're being pulled apart in anaphase. And in telophase, it gets starting to split up, and you'll see this wonderful furrow over here. It's not quite two separate cells, but it's very close. So it's not mind-mnemonic, but you could use IPMAT. I mean, it's obvious. I don't even know what it means, but that's how I always remembered it. So interphase, prophase, metaphase, anaphase, see how it's separated. And telophase, you see that wonderful furrow over there? That's what's happening there. Okay?
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Because I took pictures of mitosis, and this is one of the pictures that I took. What phase does that look like?
What phase?
Let me turn off the lights so we can see it better. See it? What phase is that? That's not telophase because there is no furrow over there. That's still anaphase. Okay? What about that one? What is that right there?
Metaphase.
Say it louder. Metaphase with an M as in Michael, right? See how they're lined up over there? Okay? So, before I give you a break, let me just show you what I did over here. I took the liberty to take pictures. And this will be on your first practical and first lab exam when we turn off the lights. But you don't need to work the microscope. All I care about is if you can identify things. I'm not going to, and I find that the little amount of time that we spend on microscopes, I find that students have more frustration about focusing and stuff, and there's so many microscope slides that we got to go through. I think what's more important is being able to identify what you're looking at here, whether a picture or something like that, than to fight with that. If you want to learn how to work with a microscope, microbiology is going to be loaded with that, okay? And they'll be spending the time for that. So this is what we call an onion root, and I want to show it now because this is all going to be fair game on the practical exam. So that's an onion root. That's at low magnification. If we get closer, you really can't see much. You get up closer, you start seeing that each one of these is a cell. And there's a nucleus in there. Now, most cases, it looks like interphase. But if we get closer, we can see some phases of mitosis. What phase is that? Anaphase. You see how they're being pulled apart? You see how you could identify that as anaphase? See how they're being pulled like this kind of thing, right? Okay. What phase is this?
Telophase.
Telophase. Why?
Furrow.
Look at that wonderful furrow. You're looking for a picture to put on your fireplace mantle? That's it. That's beautiful. When I saw that, I'm telling you, when I got a bike at 10 years old and then I saw this, beautiful, all right? So that's telophase right there, okay? Then we also, that's the onion root, but we also do the whitefish. Sometimes some of the phases of mitosis are better seen in other things. So here's the whitefish, okay? You get up closer and closer, you start seeing that these are cells all around here. So let's get up really close to it. There's one cell. What phase is that? Metaphase, right? See, they're all lined up in the middle there. You see it? Okay? What phase is this? Anaphase. See, they're being pulled apart from each other. Okay? See it? Pulled apart. So it's like, all right? So I took all pictures on here. It took a long time, but all from your slides.